Well, it looks like it's that time. Greetings, one and all. Welcome to another episode of Seminar in the Time of Coronavirus. I'm going to introduce our esteemed speaker today, even though I barely know him. So I'll just launch right into it. We're happy to have Dr. Ron Salzman of Stoller Enterprises USA presenting his seminar today. Dr. Ron received his Bachelor's of Science in Botany, of all things, from University of Wisconsin. That's the great cheese-producing state of this great union. And incidentally, Dr. Ron's favorite cheese is apparently American cheese.
Oh, what are you?
He went on to get a Master's of Science and a PhD from both from Purdue University in the Center for Plant Environmental Stress Physiology. So he's got a long history of working in plant stress, and he's going to talk about that today as well. After his PhD at Purdue, he came down and he worked as a postdoc in John Mowatt's lab for a while before being snapped up by Stoller USA, where he began as the director of bioscience, and now he's the director of research and development for the entire Stoller USA operation. And so with that, I'm going to turn it over to Dr. Ron.
Okay, thank you, Scott. Thank you for everyone for coming. And I guess that I will share my screen if I work this correctly. Okay, let's see, share. I hope everyone can see it. I'll go into presentation mode as well. Okay, can you see it as presentation mode now?
I can see presentation mode just fine.
Okay, great, excellent, thank you. Yeah, so I will talk about a subject that is probably pretty timely just now, about heat resistance to crops and the effect of heat on crops in a warming world. Here in Texas, of course, effect on our agriculture, but not everywhere in the world is as used to it or as faced with as much heat as we are here. So it does have a big impact on lots of areas of the world, and a lot of loss of yield in crops account onto heat. So I'm going to talk a little bit about some work that we've done over the last years. A little bit of the discoveries that we've found in regard to how heat works with crops. Some of this work that I'm showing, I presented last year in Europe, and that's here really high temperatures. First, but I'm sure it won't be the last. But it affects their agriculture in lots of ways. Some ways different than ours here. For example, France is obviously synonymous with wine, and you know, the French champagne here in this region has always been celebrated for its excellence. But that reputation has been built up over hundreds of years based on the cool climate of the Champagne growing region. It's very unique for that. Now, if you take a look at what happened last year in that area, as you can see, 98 or 97 degrees to 104 degrees is not a cool climate anymore. So this is seriously affecting lots of problems with their agriculture. And there's not really that many ways to deal with this problem, especially in places where they have very limited water supply, where they're growing naturally rain-fed crops and they can't irrigate. Obviously, it affects us in the US as well. There have been instances in the past, the time that I've been working for this company, that we had worked with a man named Kip Cullers, who had the record yield of soybean one year, and he had nearly the record yield of corn that year. This was in Missouri. And the next season, he didn't have a harvest. He didn't even harvest his corn. The yield was so destroyed by the hot weather that he couldn't even harvest. So that shows you can go from nearly a world record to no harvest in 1 year with corn, even in a corn producing region of the US. So the extent of the problem, it's huge, it's everywhere. Here's an article from the Proceedings of the National Academy of Sciences, which documented this program, or this problem rather, throughout US agriculture. And in this paper, they showed how the impact of temperature here on the lower x-axis, how the increasing temperatures, what effect that had on the yield, which is on the y-axis of these graphs in corn, soybeans, and cotton. And you see that when you reach a certain threshold of temperature, the yield drops dramatically in all of these crops. And in fact, it happens to any crop, and in fact, any plant for that matter. And one of the big reasons that it does happen is that critical high temperatures interfere with pollination during flowering. So we'll go into that in a little more depth in a moment, but what I could show you is that we've kind of, from the literature, gotten worked out what are the critical temperatures for these various crops. And you can see that actually these line up fairly well with what was shown in that paper. But when you reach this threshold, the pollination and the fertilization drops off dramatically. And in fact, you can also have other problems like embryo abortion that come up later, even after pollination. But this is basically showing the picture of what we're dealing with here as far as the effect of heat on yield and how it occurs, particularly in the flowering stage. This was a very illuminating article that showed the effect on wheat of not only temperature on yield, or kernel fresh weight in this case. And you see that control plants, which were grown under favorable temperatures here, versus plants that were grown under hot, sort of like non-permissive hot temperatures, 35 days, 25 centigrade nights, the kernel mass is declining significantly here. And another thing that we've seen is that in most plants that have been looked at, this one happens to be wheat, but similar things have been found in corn. The abundance of zeatin, which is a cytokinin, is actually also substantially dropped. Now, normally there is a peak of cytokinins in plants that occurs right around the time of anthesis, and these graphs show just the tail end of that. This is 0 days of anthesis, and you see the levels are high here. They actually peak right around the time of anthesis, if not slightly before, and then they start to come down. And that's a natural response of the plants during flowering. But what you see in this graph is that, again, when you have low, or rather high temperatures, the non-permissive high temperatures, you'll notice that the levels of zeatin are significantly lower here than they are in a favorable temperature situation. They both come down after anthesis, like I said, the spike, the normal spike happens sort of centered on anthesis, but you'll notice that they're already much lower here under the hot conditions. And I think the next graph actually shows that even better. This is the abundance as measured by another method, HPLC. You see that with the hot days and warm nights, the level of zeatin is much, much lower compared to the nice conditions. So I guess that makes the point that cytokinins are actually vulnerable to high temperatures. Number one, cytokinins peak around flowering time. Number two, they're vulnerable to high temperatures. And that's something to keep in mind as we go forward here. What I don't show here, but I can explain briefly, is the reasons for that are kind of known, and that is there's an enzyme called cytokinin oxidase, which actually breaks down cytokinin in plants. You know, plants have to be able to regulate their cytokinins as they want, and the way they typically do that is to upregulate the gene for an enzyme called cytokinin oxidase, and that destroys the cytokinin. But what is known, and again, I'm not showing the reference, but heat tends to promote the upregulation of that enzyme, which degrades cytokinin. So that could be a mechanism of why the cytokinin levels are down so much when it's hot. Okay, so you know, this problem is very damaging, or at least that's our hypothesis. The problem is very damaging, the problem of cytokinin imbalance. And this can have a very strong stage-specific effect at flowering and could damage the pollination and the growth of the embryo afterwards as well. So that's what we're setting out to investigate as we go forward here with this project, where I'm going to start taking up on it. I'll just mention first some of the functions of cytokinin in plants. It's named for cell division. It literally means cell splitting. That's kind of how cytokinins got their name. So they do promote cell division. They also promote bud outgrowth, or they're instrumental in promoting bud outgrowth. They are also well known to sort of delay senescence and keep leaves green longer. There's actually a phenomenon called green island, which is very unusual when a plant gets infested by an insect or a pathogen or a fungus which produces a lot of cytokinin. You actually have these so-called green islands where the rest of the leaf is actually senescing, but this one zone is staying green. And that's, I guess, known to be due to high amounts of cytokinin produced by these other organisms. Another thing that cytokinin has more recently been associated with is sugar transport. And that's the one I'm going to focus on as we go forward here. I believe that cytokinin promotes sugar transport and pollen nutrition, which helps the fertilization to happen. Okay, cytokinin and branching. This is old hat to people who are in the hormone field. Basically, indoleacetic acid, or auxin, tends to be produced in the shoot meristem, and it travels down the plant. Cytokinin, on the other hand, is produced in the roots, and it tends to move up the plant toward the bud. And there's an interplay between the cytokinin and the auxin. So this auxin tends to promote apical dominance. In other words, tends to restrain the bud, the axillary bud, from growing out. But if enough cytokinin can overcome that indoleacetic acid, the bud can grow out. So that's not the main purpose of my talk here, and we could talk more about that, but it's suffice it to know that this is a major function for cytokinin. About the senescence I was talking about, this has been done both by exogenous application, like I did here, spraying tobacco plants with cytokinins versus unsprayed, and you see how the same age plants are not senescing their lower leaves here. It's also been done by genetic engineering, where they overproduce isopentenyl transferase, which is a cytokinin biosynthetic enzyme. And this leads to overproduction of cytokinin in the plant, and it blocks senescence. So this has been done before as well. So this is another key function of cytokinin, which is, potentially very useful in agriculture, especially in areas where they do have a lot of heat and other stresses that can promote senescence earlier than you want. But again, that's not the fundamental part I'm talking about here. What I am talking about is the damaging of yield due to reproductive damage. Okay. But reproduction doesn't start when the flower opens. It starts well before that. And this graph is kind of backwards in time. This is the earliest time here. And then you go toward actual pollen shed of the flower here. But basically, what this group found with Arabidopsis, this model plant, is that if you treat the plant with heat, in other words, a hot condition, which damages it, there's only really two sort of identifiable periods during which the number of flowers or the number of fertile pods will go way down. Actually, it's the number of fertile pods, not flowers. But in other words, when meiosis is occurring here, the heat really damaged the production of pods with seeds in them. And then also at the time of pollen shed, heat also severely damaged the number of fertile siliques or pods. So the take-home message here is that flowering is vulnerable to heat, not just at one time, but at several times in the developmental stages that lead up to reproduction. So there are two potential timings at least. And then this paper didn't deal with it, but there's also an embryo stage where pollination has already occurred, but you can also have damage to yield. So this first area, again, infertility of flowers due to heat-induced male sterility. That means that pollen is not produced, for example, during meiosis, or it could mean that it's not released from the anther, which is more along the pollen shed line here. Yeah, so of course this also applies to crop plants. And the second arena, as I call it, the second time window during which heat damages yield is during embryo growth. And here's what you see when you look at a corn plant that has had excessive heat after pollination. You can see there's a lack of cell division. These kernels, even if they were pollinated, they weren't able to do the cell division properly, and they weren't able to grow, and they were aborted actually eventually. So that's another big problem. And since we already mentioned that cytokinin is largely responsible for hormonal control of cell division, it's not surprising that we can have that problem here when cytokinins are damaged by heat. Okay, so this is yet another little demonstration of what happens to corn yield and what happens to the kernel when you grow it under permissive conditions of heat, of temperature here, or in the field, that is, or in an in vitro situation. You get nice plump kernels, and they grow more or less normally, and they yield as they should. But if you grow, subject those kernels to non-permissive high temperatures, not surprisingly, this is what you see, just as we saw with those corn ears in the field. And this was all charted out and shown here how the the kernel fresh weight, the kernel dry weight, the kernel water content, all are decreased substantially by the high temperatures. Okay, again, now, so what we wanted to look at now is how does this actually happen at the molecular level? How could cytokinin work in the flower to improve heat fertility? Well, our evidence indicates that what it's doing, at least one of the things it's doing, is to promote sugar transport to the flower, to the pollen particularly. And these are needed, obviously. Flowers are known as sink tissue, meaning that they are the place where sugars that are produced in the leaves, the source, they travel to sinks, and the roots are a sink, the flowers are a sink, the fruits are a sink, the new growing point is also a sink. But those flowers need that sugar in order to develop properly and to be fertile. So the hypothesis was that cytokinin had something to do with keeping that sugar flowing. And there's some evidence to support that in the literature. The tomatoes, for example, in this paper, where they measured the sugar content of pollen under nice conditions and overly warm conditions, and they found the sugar content was decreased under the warm conditions. This agrees with the hypothesis. So this was, suggestive. So that's where I jumped in here and started out using Arabidopsis and seeing what if first of all, if I could verify these things from the literature, and then secondly, how can I investigate it from that point forward? And so I did a couple of reps of an experiment here in which I grew Arabidopsis at a partially permissive temperature. Okay, what I don't show in this slide is that I first tried growing the plants at 37 degrees C, which is roughly body temperature, day and night. And what I found is that the plants vegetatively grew okay. In fact, they also made flowers. They made exactly the same number of flowers as a plant growing under normal conditions. In fact, there was no statistical difference in the number of flowers. But what was very different is that the Arabidopsis grown at 37 made 0 fertilized siliques. In other words, the flowers looked normal, but they did not fertilize themselves, and they did not produce pods that were filled with seeds. They produced sterile, empty pods. So, okay, 37 is a little too hot. So I backed off the temperature to a partially permissible situation where they had warm temperatures during the day and a little bit cooler during the night, so I could get some fertilization occurring and some filled siliques, as I'm calling them here, meaning pods that have seeds. So what happened, I chose a heat regime where I would get some reduction in fertile siliques versus a normal, nice condition, but I wouldn't get complete abolishment of fertilization. And then, so that's where this control level is here in this experiment. Then I wanted to see if I add cytokinins or sugars exogenously applied to the plant, will it change that level of fertile siliques and how much? And so what I found is that with both 6-benzyladenine and with kinetin, which are both cytokinins, I did get, in fact, a good mean increase in fertile siliques. So it agreed with the hypothesis. And interestingly, when I applied sucrose, or this is the transported form of sugars that plants make, I also was able to increase the level of fertile siliques. In fact, this isn't a full dose response, but you can see there was a slight increase with the lower level of sucrose and a greater increase with the higher level. In fact, up to the point where they were producing the same amount of fertile siliques as the ones treated with cytokinin, and they were doing much better than the control untreated plants. Essentially the same experiment here, repeated a second time, and again the same, or at least similar response to kinetin. Well, both cytokinins increased the mean number of saliques that were filled, and the sucrose again also increased it, and again the dose was proportional to how many were fertile were proportional to the dose. So, this initial experiments suggested that the hypothesis was right, that not only kinetin could increase the reproductive success under heat, but that sugars were also implicated in it. So far so good. So we have a tentative model here of how it works. Cytokinin is what we're talking about here to start with. And as I mentioned earlier, heat stress is known to, or hot conditions are known to upregulate cytokinin oxidase. And this is, again, the enzyme that breaks down the cytokinins in the plant. It's a natural enzyme. So the idea is there that it can block, heat can block cytokinin synthesis, right? So you don't get cytokinins produced by the synthases that normally make it in the plant. So the idea was if you supply exogenous cytokinin, hopefully you can get back what you lost from the heat, and you can get the sugar synthesis, the sugar transport, and so on, until you finally get hopefully increased fertilization and increased fruit set. So that's the working model. One of the things I wanted to focus on was the sugar transporters. And I mean, I've also done some looking at invertases, but the sugar transporters were of particular interest. So what about sugar transporters? Well, it turns out there's all kinds of them in plants. And this was not the case up until relatively recently. A bunch of new ones have been discovered not so long ago, within the time I've been down here at least. So how could cytokinins increase sugar abundance? Well, if they induce sugar transporters, they can actually move the sugars around, or promote the movement of sugars around from all places in the leaves where they're produced. They have to go through cell to cell. They have to be loaded into the phloem tissue, which is the transportation tissue in the plant, and then they have to be moved to the shoot tip, the fruit, all these other sinks. But there are gates all along the way. These are the sugars don't just flow passively; they have to actually move through gates, which are specialized, and those are the sugar transporters. So this is an article. This is from an article that was published shortly after a bunch of new ones were found, but you can see that even in this article, there's a whole bunch of them here. This was earlier, when only a few of them were known. Even then, it was suggested that sugar transporters might be regulated by cytokinins, and so this was some of the tantalizing hypothetical information that made me go into this. And at the time I started, that was all the sugar transporters that were known, but then a whole new group of them were discovered, and they were called AT suite, and that stands for Arabidopsis thaliana suite, or somebody's clever idea of how to name sugar transporters. And it turns out that there's a whole bunch of them. I believe there's about 17 of them, if I'm still accurate. That's just in Arabidopsis, and these were big news when they came out. This was published in Science in 2012. And it was discovered by Wolf Frommer and his group, which are at Stanford, I think, yeah. And so anyway, they published this paper, and at the time they had just discovered all these by sequence, but there's been a lot of work since then on these proteins and how they work, where they are, and all of that stuff, what they transport, which sugars, and so on. So once that happened, I immediately said, oh my gosh, I have to look at these genes now too, to see, if I can get evidence about what happens to those with my cytokinin treatment. So first thing I did was to do quantitative real-time PCR to look at where these genes, these 17 different genes, were expressed. And what I found is that some of these are very specific to the flower, meaning the red bar is going up from the baseline here. And some of them are more specific to the leaf, which means the red bar is down from the baseline here. This is relative expression in flower versus leaf. So for example, AT suite 6 is actually more expressed in the leaf and compared to the flower, where it's much lower than the leaf here, whereas AT suite 7 is much more expressed in the flower. So you'll notice that a fair number of these are floral preference expression patterns. And for various reasons, I focused on these two, 6 and 7. So just to remind you, AT suite 6 is 5-fold more highly expressed than leaf, whereas 7 is 65-fold more highly expressed in the flower. So it's a really flower-specific isoform of this gene. And so then I looked at, well, what would be the effect of heat stress on the expression of these genes? So again, I did qPCR this time with not comparing the tissues, but comparing the plants that were grown at a permissive temperature versus the ones grown at a high temperature, which I know is going to be damaging to the pollination and the reproductive success. And so I looked at the patterns of these genes in response to heat in both the leaf and this top group here and in the flower in this bottom group. And what I found was very interesting. AT suite 6, which remember was a leaf-specific isoform, is dramatically decreased in its expression down to here by high temperatures, 35 degrees versus 24. So this one looks very stress susceptible. It looks like it's very vulnerable to heat stress in terms of its expression. This is it's a 90% reduction in its expression by this high temperature. Another one here was the same, was almost the same, almost as much. Then if we switch over to the flower, you can see that this one in particular was of interest, AT suite 7. Here we see the same pattern, whereas many of these genes didn't seem to be affected all that much in their expression by heat. Suite 7 was again dramatically decreased in its expression. It went down again, almost 90% of its expression was lost by the high temperature. So the primary hypothesis developing from this is that, or at least I'm getting evidence of my hypothesis that maybe the heat is damaging expression of these AT suite transporters, and that could be a reason why, why the plant is not getting adequate sugar to its flowers. It could be because, here these flower express genes aren't transporting the sugar into the flower, or here the leaf is not producing, it's not moving the sugar out of the leaf. That's the idea. So then the next question is, can you rescue this expression by applying kinetin, a cytokinin, to the plant? And it turns out, yes, you can. That's what I'm showing here. First, we're looking at the leaf isoform, and what we find here is that here's the gene expression at a nice temperature, 24°C. Here again is the big drop when you have the 35 degree untreated. So these are both controls, meaning there's no treatment of cytokinin, but the difference is the temperature. So again, you get this big drop. But when you apply kinetin, you can actually get some of that expression back. It doesn't get all the way up to what it was in a normal nice conditions, but it does go back up. So, and it does so at both doses. I didn't do a thorough dose response. I just tested 2 doses here, but in both cases, we rescued some of that expression. So that was encouraging. Now, if we look at the flower-specific gene, we see similar thing happen. Again, the big drop in expression of the gene by the imposition of the heat, and then the rescue by the lower dose and by the higher dose of kinetin. And in the case of the higher dose, we actually went above the level of the expression that's normal in a nice temperature. So that was more evidence that kinetin can actually rescue gene expression of this sugar transporter. So what we're really coming to here is we're approaching a potential mechanism for how kinetin can work to allow the plant to continue sugar movement to the leaves. From the leaves to the flowers and fruits. So we have some evidence at the gene expression level now for that. So next thing to do is to look for genetic data that can corroborate this gene expression data. So fortunately, we were able to get a knockdown, what's called a knockdown line of AT suite 7. That means because of all the money that science has invested in the Arabidopsis system, you can go to the Salk Center and look for mutant lines of the Arabidopsis plant, which has selectively knocked out your gene of interest. You can't always find them, but sometimes you can. And in this case, luckily I could. They have a tDNA insertion line called Salk 071508C, which has a knockdown of the AT suite 7 gene. It's not a total knockout, as it turns out, but it's a knockdown. The reason it's not a knockout is because the insertion is not directly in the open reading frame, but it's in the promoter very shortly upstream of the start sequence. So it wasn't clear. It wasn't a given that the expression would be knocked down or out all the way. So it had to be tested. So what I did is tested that by taking Columbia, the wild type in which the mutagenesis had occurred, and then I compared 3 individual plants of that Salk line, and I just basically looked at expression of that AT suite 7 under normal conditions. And what I found is what I hoped to find, which is that, yeah, this expression is much lower in the mutant line. It's roughly, on average, about a 90, in my hands anyway, it roughly was about a 90% knockout or knockdown of the expression. So, okay, we have a genetic resource that we can test now. And that's what I did next. So how we use the genetic resource is basically we want to see if we've already shown that we can rescue silique production or pod production in Arabidopsis when we apply kinetin. Okay, so now we want to see can we rescue it the same way in this mutant line as we do in the wild type. So that's what we've done here. First, in this first panel, we have no heat here. This is just standard temperatures. We want to just establish how the mutant line produces siliques compared to the wild type. And in this case, when they're not under stress from heat, the production of siliques was reasonably similar. This is not necessarily a given, but it's not impossible to explain because there are many sugar transporters, and and not any one of them may not be indispensable. Okay, so in any case, permissible temperatures, we didn't have a big difference. Now, what about high temperatures? Well, there was a little bit of a reduction in the mean, but it wasn't a significant reduction of the salic production in the mutant compared to the wild type. At first glance, that might look like, oh, that's some bad news, because it means that there's no difference, that means this gene isn't important for a production of flowers or filled salics. But remember that under these high temperatures, Columbia 0 is also knocked down per se in its ability to produce pods. So in fact, this isn't really that unexpected. In this case, we have a gene knocked down genetically. In this case, we have a gene expression knocked down physiologically by the heat. So that's not a big deal. But what the real test is, can you rescue that expression by using the kinetin like I showed before? And does that rescue operate differently in the wild type versus the mutant? And in this case, yes, it does. So you see here, when you add the kinetin with the heat, you get, this is the production level of Columbia, but this is the lower production level with statistical significance to the difference in the mutants. So this furnishes genetic evidence that that this gene is at least somewhat important for the rescue phenotype that we can show with Columbia when we add kinetin. This phenotype, genotype cannot be rescued as well. And that's probably because even though you add the kinetin, you can't upregulate this gene because it's knocked out genetically. I hope that makes sense. So that's what we found about the genetic data. All right, so that's all fine and good for Arabidopsis, but what about agriculture? What about the field? Okay, so now let's start looking at some field experiments. The first one was done by Dr. Bill Weir out in Davis, California. And he's a professor emeritus from UC Davis. And what we did here was use lima beans in the field growing in hot conditions in California. And this Excite, you see here, this is the product name from the company I work for, but it's kinetin. So consider this to be kinetin dose response. And what we found is that it worked in the field as well, and it worked somewhat dose responsively. Under these conditions, if you look at just the beans, or if you look at the beans in the shell, either way, you see that with the two higher doses, you get a significantly different and higher mean yield when you apply the kinetin to the plants during the high temperatures in the field during flowering. So this was encouraging. Now the company I work for has units all over the world, so they started testing this idea, and this one was done in Argentina, which is a pretty good corn producer in South America. And sure enough, they did 65 trials where they applied this cytokinin, and they found on average, well, they found first of all that 80% of the trials gave an increase in yield versus the untreated. And of course, there's differences from trial to trial, but the average increase was a 10% yield increase in the field. So this is looking quite useful and looking like it's fairly reliable, both good things. And it's corn, which is of course a very important crop. And as I mentioned before, it's a crop that's vulnerable to heat during flowering. Another one is malting barley. Wheat and barley are closely related, but so we did some tests. These were done in the United States through AB InBev, which is a big beer making firm, Anheuser-Busch that stands for. And we tested them tested it with them at 3 different sites: Idaho, North Dakota, and Minnesota. And what we found was that in terms of grains per head, we got nice increases in each case, substantial increases from 12 to 21% in the number of grains per head. And here's, I guess, representative pictures of what that looked like of the grain heads. And we had, better filling. You remember those corn cobs that I showed earlier, and how they had some abortion, and some of the grains were not actually developed well. That's probably what's underlying the number of grains per head being increased here. In the cases of you have suboptimal conditions of temperature, not all of the grains actually get filled, but here we get a nice increase in that. This is just a picture to show that, and it's showing the same the data that I just showed from one of the plots, the Montana. But you can see not only is there more of them, but they're also a bit plumper, and we took data on that as well. I'm not presenting that here, but again, the plumpness of the grain will be an indicator or suggestive of better filling of the grain by sugar. I mean, what's in here is primarily starch, at least particularly in malting barley, which they really prefer starch over protein. So if you're bringing sugars in here and converting to starch, you're getting bigger, plumper grains, and that's exactly what was wanted. So that's what happened there. This is actual yield in bushels from those trials. And again, you see the yield in bushels was increased in all 3 of the trials with statistical significance, in fact, in all 3 cases. And again, very nice increases between 15 and almost 19% in bushels. So this is a very useful, very, very desirable type of increase, and it's also been consistent across all these trials. So this made them very happy. This is some data that I just got back. It was actually, the trial was done in 2019, but I just got it back fairly recently. This is a trial I did with, I collaborated with KWS, which is a big seed producer that's in Germany. It's actually the 4th largest seed producer in the world, I guess, according to Wikipedia. And what they make a lot of different types of seeds, but in this case, we're looking at beets. They actually approached me. One of these guys, this guy here, Olaf, saw a poster I had presented at an academic conference, and he was interested in talking. So we got together and did discussion, and they were interested in setting up these trials. So they did these trials, they set them up, they did everything. I just sent them the product, and they did the applications and took all the data and all of that. So this is their data. But again, the data are very, let's just say they were pleased. In 2 different breeding line combinations, we're just calling them A times B here and C times D. Their goal, remember, is seed production. So since they're a seed company, and so they're interested in large purified seeds. So that's what these numbers are showing. The treatment gave in this hybrid, in this particular case, it gave a 72% increase in this large seed category. This one was not statistically significant, probably due to variability from a relatively small N number. But in the case of this other combination, even though it's still a small N, they got a significantly different increase in this case, and it was even larger. It was 119% increase. This is it's more than I would have expected, but I'm not complaining. So that's what they found there. And the other thing they told me to look at, they sent me the data and they say, calculate the number of seeds produced. I think you'll be even more happy. So I did that, and that's, yes, it is even more dramatic. So this is the actual number of seeds that were produced. The control here and the treated, there was more than a 3 times greater number of seeds produced, and that's what they sell. That's their money. So needless to say, they're interested in continuing with this, probably into a commercial scale next. Similar thing in the other hybrid, in the other field, 125% increase, again, statistically significant. So this is, I'm not saying this kind of thing happens every time, whenever, whatever crop, usually it's not going to be this good, but for them, hey, they were happy. So that's what can happen when all you have to do is make seeds. All right, so I guess I've gone through most of my slides here. This is just a recap. You know, what I think is now I can understand at least part of the mechanism of how heat resistance happens, because the cytokinin can promote gene expression of a key, at least one key gene, and perhaps more, the sugar transporters, which allow better sugar movement to the flowers when they need it, and better nourishment of the pollen. And how does that happen? It appears that this kinetin treatment can restore gene expression to the gene that's normally knocked down by heat because of the cytokinin oxidase effect. But we substitute this the output of that gene back, of the cytokinin synthase back in by feeding it exogenously. And then we let the thing carry on like it should be. And of course, we've got statistically significant and economically important increases in yield. So that's kind of a beginning to end story that how you can take academic research and turn it into results in the field that work. So I guess it's been kind of a good story for us. I just want to acknowledge people who contributed to this. There's been a number of people in the US, in Argentina, also Chile, which somehow isn't on this slide. I don't know how that came off, but it's Chile has worked with us as well. Mexico has, South Africa, and then Stoller Europe, and also KWS, who I just mentioned. So all of these people were involved in various ways. And okay, I guess with that, I'll call it a day here until I'm ready for questions. So I thank all of you for your time.
Great job, Dr. Ron. Great job. Impressive results. Questions?
Okay, thank you for the presentation. Just like the way I saw and the way you explained the interaction between the kinetin and the sugar transportation.
Mm-hmm.
I also have some knowledge that there is like gibberellins can promote flower production and fruit production. So, but I'm not so certain about the involvement in the sugar transportation and all those other stuff. But during heat stress, can you administer gibberellins? Can they help like to promote the flower growth and the subsequent fruit production?
Very good question. I'll just be short and straight to the point. I haven't looked at gibberellins, and at this point at least, I don't know of anybody who has checked that. However, I have seen, during the time I was working on this, I have seen a paper, actually I think it was either Nature or Science, but it was quite amazing because they got a paper in one of those top journals basically showing nothing more than how auxin was doing this, essentially the thing, it was promoting better heat resistance in terms of producing the yield, producing the successful fertilization. Okay, they didn't even have a mechanism particularly, they just had They just had pharmacological data from treatment with the IAA, and they had a mutant line, which was an auxin biosynthetic mutant. And they showed with both lines of evidence that supporting the idea that auxin can also play a role. Okay, how the role works for auxin, they didn't actually know. So, you know, that's not an answer to your question about gibberellin, but it is another plant hormone that has at least some role in you know, tolerance to heat for reproduction. Gibberellins would be another one very interesting to look at for sure.
Hey, Amir, why don't you ask your question?
Yes, Ron, a lot of the data you showed is heavily focused on flowering.
Yeah.
But heat stress can actually happen at any stage in the development. And if it happens early, it can affect the plant height. It can cause height reduction. It can cause lower tillering ability. So is that application going to help when you have issues early on in the season? That's one question. The other question is, you know, sugar transport is also part of male sterility, which is used in hybrid seed production.
Yes.
So is that the same mechanism, same pathway?
Okay, so let's answer the second question first. Yeah, I didn't include it in this talk for time purposes, but yeah, of course you're right. Actually, there are cytoplasmic male sterile lines which are used by breeders that have overproduced cytokinin oxidase. They know exactly how it works. They know that this line is sterile because they were overproducing cytokinin oxidase, and there are pictures of this in academic journals where you can see the night and day difference. There's no pollen on the anthers in these lines because the cytokinin production is blocked. But when they supplement even these sterile lines with exogenous cytokinin, there comes the pollen. You know, they can produce it. It's just that they can produce pollen. It's just that they can't produce their own cytokinin. So yeah, now as far as is it the same mechanism of sugar transport? You know, obviously I haven't tested that, but I would suggest that it might be, yes. But the sterile lines is blocking the production of cytokinin per se. So, you know, I've shown one thing that cytokinin can do, the sugar transporter. It may be able to do other things for reproduction as well. So back to your first question about the other times in the developmental cycle. So this is maybe a bit more complex. Heat, you know, in this case, heat and water are often going hand in hand as far as plant success and growth in earlier stages. Obviously, heat requires more or induces more transpiration. So the water use tends to be higher. If the plants can't get enough water in the roots, obviously that's going to be a problem. Heat, like many other stresses, can also promote ethylene production, and that is another hormone which is not necessarily directly, it may or may not be tied to cytokinin in any specific way for this purpose, but I don't know of, other than general nourishment of the plant with sugars, and of course, as I talked about, induction of branching and cell division. But yeah, I mean, I guess those are pretty important for development, right? Cell division. So even vegetatively speaking, if cytokinin expression is damaged, yeah, that could be a very serious problem for even early growth of the plant. Yeah.
Okay, Jennifer?
Hi, yeah, so I was wondering, in the beginning of your presentation, you mentioned that like auxin works top down and then the cytokinin, they work from the roots up. So I'm just wondering if you're looking at soil temperature or air temperature when you're doing your evaluations, and like if there even is a big difference, and if there is, would that change anything?
Yeah, good question. In the case of the Arabidopsis work, these plants were in an isolation box, a temperature-controlled box. So in that case, the soil temperature was basically equal to the air temperature, because the pots were very small and they're not a heavy, dense soil. So they basically don't have any buffering, or very little buffering for temperature. And they were probably following, the soil was probably following the air temperature. What I measured was air temperature, but I think that for all practical purposes, the soil temperature was pretty close. Out in the field, of course, in these various places, things could be different. It depends on many factors, whether the soil temperature got really elevated or not. So, I can't answer that question, but air temperatures were measured in some of these field experiments. So at least we know what those were, and that they were, getting into the area where it was damaging, based on those thresholds that I showed earlier.
Kirti?
Nice, very nice talk, Ron. Question. I have a couple of questions. One is regarding the way you treat, especially in the field, and it's just a simple spray, and then I guess the stage is pretty important. At what stage do you spray?
Yeah, that's right. So yes, it's a simple spray, a foliar. Well, it's a foliar spray, but try to focus on the flowers if you can. But yeah, just a foliar spray with a little bit of surfactant, and the timing. What we've been doing mostly is to do, for example, in the beet trials, we did one spray just as flowers were starting to open. And then we followed it up with another spray a week later. In other trials, we generally have again focused right around the anthesis point. There may have been one trial where we did an earlier treatment as well to try to catch that meiosis vulnerable spot. But in general, we've focused around flowering mostly because it's very easy for people to to recognize, okay, it's time. I can see the flower buds, I can see the flowers are at 10% or they're almost starting, and it's easy to focus on that, and it generally has worked for us. I would also mention that we've found that even under conditions where there isn't too much heat stress, we actually have been getting very good increases in yield as well. So, just that's separate from your question, but it actually has shown pretty good results even without heat. But yeah, I would focus right around the flowering, the opening, just prior to opening, and probably right after for another one.
So one of the reasons why you get big yield increase as well is it because you got more green leaves now because of the cytokinin treatment? There are more photosynthetes going up to the seeds.
That's a really good possibility. To be honest with you, we haven't monitored that in these trials. We've been focused just on looking at the reproductive side of it, but very, very possible. Certainly that phenotype is known. So right, and cytokinin is actually known to stimulate photosynthesis, as you probably know.
Yeah.
So yeah, it's very likely part of the picture as well. Yeah.
Thank you.
Hey, Jody wants to know, how much does it cost to apply?
Well, I'm not in the sales part, but it's perfectly affordable.
When I buy Kynotin, it costs about $40 a bottle from Sigma.
Yeah.
Can you get it for cheaper?
We'll give it to you at a steal. We're an agricultural company. We're an agricultural company, so we charge less than Sigma.
Okay.
I have good margin.
Sorry, Ron, this is Eugene. I have two simple questions for you. You mentioned a little bit that the heat and the water go hand by hand. So in your greenhouse study and also the field study, did you control the water? So actually you don't have any water deficiency issue or not? This is my first question.
Yep. So
Okay.
that's, yeah, we made sure they were well watered. So
Okay,
all
great.
of the, yeah.
All right.
Mhm.
Second question is related to the application. You mentioned a little bit that you use the surfactant here. I'm wondering what's the half-life time of the what's the name of the cytokinins on the leaves? How many days or weeks do they last?
Yeah, I'll just be straightforward. I can't answer. I don't have the answer from our own studies. We haven't tested that. It might be available in the literature. I don't really know, but in general, those of you who work with hormones, plant hormones, know that they don't hang around forever when you apply them exogenously, and some more faster than others degrade. So I would say, just as a rule of thumb, we try to think in terms of 5 to 7 days as the timing after which we don't have much of an effect from that application anymore, or it's a nice rule of thumb, repeat on a weekly basis if it's something that we want to apply repeatedly.
Okay, great. So if you want to optimize your formula, we may help you a little bit as work together to see if you use some clays to work for you. Maybe if you want to make it last longer or shorter. This may be something we can discuss later. Thank you. Very nice presentation. Enjoyed it.
Thank you.
Mr. Fonseca, you got anything?
Yes, I have a question. Thank you very much for your great presentation, Dr. Salzman. But I was curious about when you start talking about hybrids, and it was a great increase. And usually when we talk about abortion or male sterility, we're talking about the male side of the seed production. So when we are applying your product, we usually apply it on both plants, the males and the females, the same amount. I was curious about if the amount of the application would change if you changed the male or female plant.
Yeah. Well, in the beet study, if I remember correctly, they applied it on both lines. Well, actually, yeah, I mean both breeding lines. They applied it on both the male and the female side of the cross. And I guess, your point, of course, is, if we're talking about male sterility, obviously it's important to put it on the male. But, we also wanted to put it on the female because, there's the potential for abortion that could happen later in the female parent. And we wanted to, catch that possibility for improvement as well. So, yeah, we could have done it, only looking at the male. But this was sort of a first field study, and they just wanted to see if it would work, throw everything at it and see if it would work. So, yeah, we used it on both sides. All right,
and the amount were the same?
Yeah, the amount was the same on all plants. Okay. Same dose. And it was a easily affordable commercial dose. We didn't do some crazy high dose.
Thank you.
Any other questions?
This is Kirti. I have one more question. One thing that I was wondering about in terms of pollen and pollen tube and so on. So obviously pollen needs sucrose, just for development and everything. But once it's formed, it has to grow all the way down through this stigma and style. And so the availability of sugar in those tissues is pretty important too.
Absolutely. Yeah, absolutely. You're right.
Okay.
Yeah, so that's, I mean, it can help both sides of the equation, I guess. If the pollen is better nourished when it's produced, it'll have more, initial reserves, I guess, for its germination. And then, like you say, the female side will be better off nourishing it if it has more sugars as well. So you're right. Yeah.
Okay.
Any other questions? All right. Well, I'd like to thank you again, Dr. Ron. You did an excellent job, and
thank you.
See you in the lab at some point in the future.
All right. Very good. Thank you, guys. Thank you all.
Thanks, Ron. Appreciate it.
Okay.
Great job.
Bye-
Thanks.
bye.
Next week,
All right.
people.